Hello, everyone, and welcome to today's DFHCC Connect Science seminar. I'm delighted to introduce today's speaker, Dr. Fred Alt. Fred is the Charles A. Janeway Professor of Pediatrics and Director of the Program in Cellular and Molecular Medicine at Boston Children's Hospital. He's also a Professor of Genetics at Harvard Medical School and a Howard Hughes investigator. Fred has a storied career with a continuous stream of important discoveries related to double-strand break repair and genomic instability, and his discoveries have been recognized by numerous awards spanning several decades. Fred told me that he's most proud of the William Silen Lifetime Achievement for Excellence in Mentoring Award, which he received from Harvard Medical School in 2016. A major focus of his research across the years has been on mechanisms underlying programmed rearrangement in immunological development. His research placed this mechanism on the map in relation to now known pathways of double-strand break repair, and in recent years he has extended this to understanding the relationship of these processes to higher order chromatin structure, which we'll hear about some of in his presentation today. I'd like you to remind you to consult the Connect Science website for forthcoming seminars and for recordings of previous seminars. Secondly, please be sure to write your questions in the Q&A box, not in the chat box. Your questions will only be used if they appear in the Q&A box. So please do that. So without further ado, I'd like to welcome Fred, whose talk today is entitled Modulation of Cohesin Complex Factors Regulates IGH Locus Contraction and Long-Range VDJ Recombination. Fred, I think you're muted. Fred, you're muted.
You hear me now?
Yeah, it's good.
Thanks. I'm not sure how I got muted. Well, okay. So here I am. And I'm sorry, I think I missed the last one due to something that was unforeseen, but I'm really pleased to be able to give this lecture again, just about 1 year after I gave it before, last year in the Jimmy Fund building. Tom Look hosted me for that one. So I'm going to update the story I told then, which is a story, it's sort of building on a story I've been telling for about 40 years. And I think we're not getting to the end of it, which is good. So as Ralph said, the title is The Modulation of Cohesin Complex Factors and how modulation of those factors regulates IGH locus contraction and long-range VDJ recombinational scanning. And that is new from last year. So to give, I don't know if you can you see my pointer at all? I'm moving it around there on this whale tail.
Yes, we can see it.
You can see it? Good.
Yes.
Okay, so this is a tail of a whale that is going under our boat a few years ago that one of my postdocs got inspired by and put his favorite molecule on the whale tail, which is a subunit of an immunoglobulin, which consists of 2 identical heavy chains and 2 identical light chains. And the N-terminal portion of these chains is the well-known variable region. It has a variable amino acid sequence, and it's assembled from germline V, D, and J segments for the heavy chain, and for V and J segments for the light chain. I'll have something to say about both. Very little about the light chain, but a little bit. Now I gotta get this thing moving again. Doesn't do it by the normal process. Okay, so let me tell you what VDJ recombination has been thought to be for many years. It's been thought of until just the last few years because we just didn't know enough to think about it differently as a very linear process. And it's a process in which, for example, a D or a J get joined together to make a DJ, could be a V joining to a DJ. And the way that works is that J's and V's are flanked by something called recombination signal sequences. I'll tell you more about that in a minute. And those are targets for binding to an enzyme complex. It's called of RAG1 and RAG2, which is the RAG endonuclease. Sometimes people call it a recombinase. It's not really that, it's an endonuclease. And its job is to recognize those sequences, make double-strand breaks between them, and then those breaks get handed over and get joined by a pathway that we had a major role in discovering, one we call non-homologous end joining, or we've named it classical non-homologous end joining to distinguish it from other end joining pathways. And that puts the ends together. So that's pretty much the basic activity that is involved, but it's far more complex than that for a variety of reasons. One being it goes over megabases of chromosomal distance. So there's a lot more going on there than that linear type of reaction that you might see in a plasmid reaction. So how do we know now much more than we did a few years ago? And the reason is that we spent a lot of time developing high throughput assays to study this process and some other processes. And all of these assays are based on an assay that we published about a decade ago, or more recently in a revised form, in an upgraded form called LAM-HTGTS, or linear amplification mediated high throughput genomic translocation sequencing, which is a method that works very well for many different things. And it worked very well, what I'll tell you here, for turning it into a VDJ recombination assay, because we can make any VDJ introduced break our bait and find out exactly what it joins to and how frequently it joins, and do lots of other things like that. But it's far more sensitive than any prior assay, maybe 100 to 1,000-fold more sensitive. So that allowed us to see things we just simply couldn't see before. I'll tell you more about that. And we also developed it into, over here, this 3C-HTGTS, which is high resolution genomic sequence interaction. And basically what that works out to be is just a much higher resolution assay to do what one might do with a 3C-seq or a 4C, but really a high resolution. So it's been very helpful for us. Okay, so that's all we've turned into other things, but not for this studies. So now I want to just remind you about how chromatin is organized, because that's critical for what I'm going to talk about. And particularly, you all know that chromatin is organized into domains of various sorts, but a large fraction of the domains in the chromatin are loops. And the characteristic of many of these loops is that they're bound by the CTCF binding factor, and it binds convergent CTCF binding sites that I'll come back to. But CTCF is an 11 zinc finger containing chromatin architectural protein. It binds directionally to a 14 base pair core factor binding element. It's been around. Its name is something that's not what it was named for. What we call it now is not what it was really named for. It doesn't matter. But eukaryotic genomes contain many megabase or sub-megabase contact loops. In other words, these types of loops that have contact at the bottom. And at the bottom of these are pairs of CTCF in convergent orientation. So with respect to the sequence, they point at each other. So that becomes a very important point later in a second. So now I want to tell you a little bit about what we know about VDJ recombination. First thing we've known, we published a paper about 10 years ago that they find an element. I don't know why he's moving around, but we know that VDJ recombination takes place in convergent loop domains, but not necessarily CTCF bound. They're different. The one that is CTCF bound is one down at the downstream end of the locus. It contains the constant regions that undergo a totally different type of recombination reaction that we've also recently defined to use loop extrusion, as I'll tell you. Then it has 4 J segments, all flanked by the recombination signal sequences that I mentioned to you. And then about 8 or 9 Ds, depends, this is in mice. And then an element we call it IGCR1, which is defined by 2 divergent CTCF binding sites, one pointing at a bunch of sites that are downstream of the locus as a loop anchor. And then further upstream in the locus, over the next several megabases, there are hundreds of Vs that can be joined to the Ds. And those Vs in their proximal regions are all flanked immediately by CDCF binding elements, and the more distal ones, they're interspersed here and there. But what I want to get through is the VV locus, 2 megabases or so of it, is just interspersed all throughout it with many, many CBE elements. It's very different than most parts of the genome. That's because these are really critical for its regulation in a way one might not expect. They do more than one thing, as I'll tell you. So let me also, because it's critical to understand what we're looking at, that these arrows, these recombination signal sequences, consist of the coding region, say, of a D or J, then the sequence CACA-GTG, that's highly conserved heptamer, a spacer of 23 base pairs, and then an AT-rich nonomer. And then it goes into the intronic region. So there are 2 forms of these, one with a 23 base pair spacer and one with a 12 base pair spacer. So now, a point that I'll get to in a minute is, while this locus is very large, this several hundred base pair domain at the downstream end has the Ds and Js, and that's secluded from the Vs by this IGCR1 element, and it allows, during development, something we've showed a long time ago and more recently, the Ds join to the Js to make a DJ intermediate before a V will ever join to the DJ intermediate. V doesn't join to the D by itself. And large part of that is because of this element that puts them into a different domain. So now the key point here, as I mentioned, so the first step, the D to J joining part, all goes on in this one loop domain that's bounded by the IGCR1 and really by something called the recombination center. Lots of things to remember, unfortunately. So what's the recombination center? The recombination center is somewhat what it sounds like. It's the J's, a couple of enhancer elements, and a most proximal D. And when RAG protein binds to that, which I'll show you in a more easy to gather form, that makes it a recombination center. And it sets up to recombine in an orderly and a very specific way with everything that's upstream of it in this case, because the J's are pointing that way with the combination signal sequences. So now getting below, we get to this, the RAG structure, which is actually very critical for understanding how this all works. And this RAG structural work was done somewhere down here. It might have got, some of it might have got, oh, it's on the next slide. I put it on because I wanted to highlight it, yes. So the RAG structure was done by Marty Gellert and Wei Yang in several papers, and also by Hao Wu's lab in several papers recently. And knowing how RAG binds these recombination signal sequences is really critical to figuring out how the whole thing works in a larger scale. So again, what you have to have matched here are sequences with 12 and 23 RSSs. And I go back to this, the J's all have 23, the D's have 12s on both sides, and the D's all have 23. So D's can join to the J, in theory, in either direction, and the V's can join to the D, to the upstream DRSS. And in theory, they could join to the J, but they're prohibited by this element in between. So you have to have a matched 12 and 23 bound in a very specific orientation to the RAG, which is, I don't know, you call this a heterotetramer or different things, but 2 molecules of RAG1, 2 molecules of RAG2, and the catalytic site is in RAG1. So when these 2 elements bind, they have to be bind so that these 2 recognition sequences with their CACs going in those directions face each other so that the RAG catalytic activity can cleave them. And RAG can cleave something if it's against what we'd call a canonical RSS. It can cleave something as simple as a CAC at a very low level. But that is something that allows us to see where RAG goes on the chromatin, because we can follow where its tracks are by cleaving those kinds of short sequences that are very frequent. So that brings me to this other point of background. What we found a number of years ago, Jiaxi Hu and Yu Zhang in the lab, is if they took something the equivalent of a D and a J and an enhancer, so to make an ectopic recombination signal sequence, and they put 2 of those in opposite, with the RSSs in opposite directions, in various domains across the genome, using our high throughput assay, we could find that these ectopic recombination centers in convergent CBE-based loop domains support RAG scanning directionally over the entire domain. So if RAG binds to this RSS, it will look this way up to 2 megabases at least on the chromatin for these cryptic RSSs, and it only uses them if they're pointing at this RSS. So it scans directionally. They have to be in convergent orientation to be used. And as I said, it can join hundreds to thousands of these as it scans over a megabase or more, a couple of megabases of DNA. The only thing that will stop it is when it gets to convergent CBE loop anchors. That sounds familiar. And then it stops. And then if you delete those, it continues to go further till it hits the next one. So it's being withheld in these CBE-based domains. So we proposed at that time that this exploration could be driven by COHESN or transcription. We didn't really know. I mean, we knew it was something that had to do with COHESN probably because it was in these domains that are bound by COHESN and CDCF. But then the real thing that got us thinking in the right direction came along from Lieberman Aiden and Leonid at MIT, Mirny. And what they noted was that CTCF site binding sites sit in all orientations in the genome. They can be convergent, divergent, the same orientation, but noted that they're always got joined together into loops, not always, but predominantly from all of their, analyzing their high C data when they were in convergent orientations, and one that might be, for example, in the same orientation would just pass through the loop, not making as strong an interaction. They do have interaction though. So they called this process loop extrusion. So the process is based on a protein called COHESN that has been around for a long time. In favor of Kim Naismith and people like that, and his proteges for over the many years. It's a highly conserved chromosome multi-subunit ring complex. It's required for sister chromatid cohesion and also for chromosome segregation. And it's also been implicated in gene regulation in various contexts, too many to mention. But COHESN was thought, according to this model, to be the driver of chromatin loop extrusion. And that was actually debated for many years. I mean, if this is really the model, I think one thing that solidified it in this context was that two labs recently showed that COHESN, this complex of SMC, SMC1, and RAD21, the one we'll focus on. It's a key complex that holds them together, and it's responsible for regulation of the complex. They showed this complex can actually extrude naked DNA in vitro, showing that it has the right activities. And in vivo, it's all been, a little bit correlative, but Chao-Ching Ba in my lab did deplete it in vivo and used very nice controls and showed that it could knock out loop extrusion in many different contexts. So I think everybody pretty much now feels confident that COHESN is indeed doing what it was proposed to be in this model. And for us, RAG scanning and loop extrusion rang a lot of bells because they're both linear processes. They both employ the convergent rules. And they're both restricted by the same loop anchors, which the major ones that people know about are CBEs. But loop extrusion itself can be slowed down by lots of things, by genomic structure, by transcription. We've shown by dead Cas9 in its pathway. Always, both processes get affected the same way, and some of it's in these papers that are below. So then we started to try to pick it apart about how, since we believe this process is working based on all the evidence we had, how is it being harnessed to work in the right way for all the things that are going on during VDJ recombination across the 3 megabase IGH locus. And the first set of experiments I'll tell you about very briefly. They've been published recently, last year, or maybe the year before, I guess now. Yu Zhang and Sufei Zhang. Yu's now in Michigan with her own lab, and Sufei will be starting his own lab at Peking University in about a month. It's sad to lose both of them, but they'll do a great job. So every study I'm going to tell you about until I get more towards the end is using something that we and many others call V-able transform pro-B. Some people call them pro-B cell lines or previous cell lines, but early developing B cell lines. And I'll tell you why they're a little bit of a confusion, at least the way we study them. And I've been working with these lines for, I think, about 40 years. And I think David Baltimore once said, Abelson lines don't lie when you're thinking about what goes on normally. And I think as long as you look at them in the right context, I mean, they don't. It's quite amazing. You'll see what I mean when I get to the end. So, you know, you get mice and you whatever type of mouse you want, and you take out the bone marrow and you transform them with the retrovirus, or you put BCL2 in them because later on when you want to treat them with an enable tyrosine kinase inhibitor to arrest them in G1, you don't want the impact of arrest to kill them, and BCL2 keeps them alive for days. And then you can do all sorts of experiments in them. And so those are the lines that we use. And you know, the people going back to Naomi Rosenberg and Mark Schlissel and many others and Barry Schleckman, who developed a lot of details of these systems. So now when we G1 arrest a particular Abel line, and those we can make any kind of a mutation we want in them pretty much, particularly with Cas9 guide RNAs, you can just pretty much do anything you want very quickly. They have, and the wild type line are extremely interesting, and this becomes sort of a real punchline for the story when I get to the end of it. They have two loci: heavy chain, light chain. Heavy chain locus, D to J, V to DJ. Light chain later in development, as we showed many years ago, and that does just V to J. And both loci are very large, multiple megabases that they got to work on. Okay, but we focus on the IGH locus, and what's really interesting about Abel lines is they once you arrest them, and when you arrest them in culture, they will express the RAG protein, and once they're arrested in G1, and will undergo really robust D to J rearrangement, but they never touch their V's. And the reason is that they don't have something called locus contraction, and I'll get to that later. So, but on the other hand, they fully rearrange their light chain loci. So they're sort of a little bit schizophrenic in the sense that they normally would do D to J, then contract their locus, and then go on to do light chain. But these guys, D to J, skip the contraction, so no V to J, and go on to light chain. So they make like the absolute perfect thing if you want to figure out what is controlling locus contraction, and you go back and figure out how can you induce it in an Abel line. And that we've done, and I'll tell you what that's about. But the D to J part is I talk about quickly, but it's I think Yu Zhang and Sufei Zhang did an amazing story here. So Ds lie upstream of Js. They have recombination signal sequences on both sides, and the Vs lie further upstream, excluded, and they have recombination signal sequences pointing at the upstream signal of the D or the J. So it's known that D to J joining theoretically could go one way, two ways. Either the downstream RSS of the D could get involved, or the upstream RSS could loop around and get involved in the right direction. So you could get joining of either deletion or inversion. In plasmids, in fact, you see both, and that fooled people a lot because they thought the DRSS must be somehow telling the cell, because most cells, 1,000 to 1, join deletional D to J joining over inversion, and they don't hardly ever form by inversion. As I said, in a plasmid, you get a different answer, but chromosomes aren't plasmids. So the prior model was there's differences between these two DRSSs. Our model was it would work with just RAG scanning. So Yu Zhang did just a huge amount of mutagenesis of Ds, turning them around, different orientations, turning the whole locus around, turning one or another around, And, you know, a huge amount of work. And what the work led to, along with other things, is the model you see here. So the way we believe it, and I'm going to show it to you a little easier to see as an animation. So RAG binds to the recombination center, that pink, which includes the J's, and it also includes 1D, which is great because that D it's so close it can access RAG by diffusion, which means it doesn't have to access it by scanning. So, but it actually gets pulled out frequently, and then RAG continues to extrude, and it extrudes D's past the recombination center, and it's bringing them in such a way that it can see their convergent RSS, but not their divergent RSS, or RSS in the same direction. So those don't get rearranged. If you invert them, it does just the opposite. It will rearrange the one that you've put on. It'll rearrange whichever one is pointing at the J. Until it gets up to the end of this, you get this thing called the DFL16, which is the most used D. And that gets used very frequently. And that's because it's blocked right here at this IGCR1 CBEs, 3' CBEs. It just can't get past that block. So RAG gets lots of time to focus on it. So if this is just to show you what this looks like to scale. I mean, to scale in terms of how big RAG is, which always never ceases to amaze me, or David Schatz, people like that. So you got these two CBEs, you got the Ds, or these orange, green, and white in between, you got the Js. RAG binds to the recombination center. This is the true size of RAG relative to, say, a COHESN molecule, which has great implications also because it impedes scanning, and it impedes both directions, but one more than the other, which has implications later. So now you have RAG stuck here. This end's not stuck. RAG and the recombination center, so you don't go that direction. So now COHESN starts pulling the loop through, and it pulls the Ds through, and at some frequency, when they're lined up correctly, it's like that, they get used. And it will do that all the way through, so you see only the convergent Ds until this whole complex gets up to the CBEs. And then that FL16 shows just what the reaction looks like. They get cut and joined. And that will happen at varying levels for the other ones. And there are other things that can make them keep going or stop. You know, again, this is a very simple-minded version of this, just like the versions of loop extrusion are, but it has to give you a place to begin to work and think about how you go to the next step. Okay. Now, the other thing that Yuzhang did, and mention this quickly, is the orientation of the recombination center, or the RSSs in it, is what tells RAG how to scan. We knew that from our experiments of putting recombination centers across the genome in different CBE loops, but we did it here because she wanted to go further with her study. So when the DJ is pointing upstream, like you've just seen, the scanning will go of upstream chromatin past it. But if in the same paper, when she took the DJ and inverts it, so now the DJ is pointing downstream, it binds RAG the same way, but it came in from the back this time to an active site, but now it will scan the chromosome that goes down the chromosome. So now it will continue on down the chromosome, scanning chromosome in the opposite direction until it gets to the 3' CBEs, that anchor. And in there are all the constant regions and other things that we can manipulate. We can make them transcribe. So we did a lot of things because there are no RSSs here, nothing to stop it. So we can just see what stops RAG scanning. And we found what does are CBEs, for sure, that's the biggest one, but transcription, active chromatin. And what we're also able to do was to make catalytically dead Cas9s that would bind to these repetitive switch regions and show that that would really stop extrusion very well. And some people in the field have been trying to do it. I think you had to have enough target sites to make it work. And what it showed, because every time we put something that would stop RAG scanning, it would focus on cryptic recombination sequences under the sequence that was impeding it, which shows us a mechanism for how transcription really focuses what we called years ago chromatin accessibility of RAG. It's basically by keeping sequences together to give them time to find each other and actually work. Also reveals how off targets are getting into RAG complexes across the genome and translocations in progenitor lymphocyte cancers. I think really explains a lot of those. Okay, almost getting to the more recent interesting part. So one other thing we did, work done by Suvi Jain and Xiaoqing Ba, we asked what would happen if we deleted IGCR1. You know, so now scanning could go beyond IGCR1 and it would get up to the V's. So when we deleted IGCR1, indeed, IGCR1 it allowed the chromatin to be scanned upstream beyond the D's from a DJ, and now to reach the proximal V's. There's a most proximal V that we pointed out about 40 years ago, George Yankopoulos and I, as being, because we'd actually proposed scanning in a 1986 paper. Nobody believed it. We're both happy that it ends up being this way. So this VH 5.1 is different from these other ones because it doesn't have these additional dots on it. The additional dots are CBEs, so or the purple things behind the dots. And so basically all the proximal V's have CBEs. So extrusion goes past that 5.1, but as soon as it gets to the VX that I cloned years ago, even more years, called VH81X, the most proximal V, that V gets used the vast majority of rearrangements. It doesn't go much further. It goes a little bit to the next one, a little bit to the next one. But if you mutate the CBE, then what happens is it goes to the next one and it becomes by far the most utilized. VH81X no longer gets used, and same thing to the next one. Then the real killer experiment was Suvi noticed that this one that we said had to be different in our model because it never gets used, even though it has a perfect RSS. It doesn't have a CBE. It had a vestigial CBE. So she mutated that back into a full-fledged CTCF binding CBE. That thing goes, nothing else gets used. It goes, you know, so it really proves that the CBE is important. So for these proximal Vs, the CBEs can pull them into the complex and really allow them to get rearranged. And they're doing that in a very positive way. But what they also do is nothing gets past them. And then you remember again, up the locus, hundreds of CBEs. So if I told you, you know, RAG scans the whole heavy chain locus, I mean, I told myself that, how in the world could it do it? I mean, because you've got all these impediments, you know, transcription, CBEs, how does it get through? There was a model put out by a few different labs, Busslinger, James Scott, Case Murray, I think it's from his paper. What they found years ago using microscopy first was that the IGH locus somehow, instead of being this nice linear thing, appears to contract. And the idea was it contracted into these sort of ringlets that would all go around, be close to the DJ, and then by diffusion, it would equal the playing field and V's could get in. And that was a model that also in retrospect could say, that's how you could get around CBEs also, because if you came in by diffusion, the CBEs wouldn't have any role. You wouldn't have to move linearly past them. So we decided to test that. And the way we tested it is that we found, again, this whole thing about cryptic CBEs, CACs. So they exist in both directions on both strands of all these loops. And so if this model of diffusion was right, you would say that you would only use the CBEs that are pointing in here in red at the same pointing, you know, convergently to the DJ. And these other ones on the other strand of the DNA wouldn't be seen because they're a GTG, they're in the wrong direction. In this model, they both get used because they both diffuse in. So that was what we tested. We started in Abelson lines, Xiaoqing, Jingman, and Adam are by the guy who does all the computational biology. This has also been published fairly recently, this past summer, I believe. I'll mention it quickly, but here's the point. I want to make sure I get everything done. I'm doing okay. I only got about 10, 6, 7 slides left. The IGH locus, as I mentioned earlier, I just like to emphasize these things because it takes a while and all these things for all of us to get it down. They're not contracted in these pre-pro-B lines. They've skipped it. And you can see that because if you do HTGTS in a bone marrow pro-B cell, and you look for what rearranges. What rearranges first, this way down on the box of the locus, all the Ds. And then you go up the locus over the 300 or 3 megabases, and the hundreds of Vs all rearrange, albeit different frequencies, but they all rearrange, very reproducible. If you go into a V-able line, you see what I just told you, the Ds rearrange like crazy, but only a little bit of leakage to this proximal 81XVs and then nothing upstream. So it's also true using his high C assay. You can say, what about locus contraction? So in the bone marrow, you see the thing we call the recombination center. It interacts with a number of things, 3' CBEs, other things, but it interacts with very discrete pieces of DNA or chromatin across the IGH locus. And that is sort of a hallmark of what you would call locus contraction. The whole locus is interacting. In this case, other people like to say more with the CBEs, but we argue with the recombination center, I think, and a lot of arguments for that for many papers. In the able line, there's a little tiny bit of interaction, but very, very little. And we'll come back to that. And we know from things we've found years ago, if you use growth-seq, the distal VHs are really transcribed at a high level. The proximal ones, particularly the ones that have CBEs, are not. So the question is, what do you have to do to get this to behave like this? In other words, get the bone marrow probe line to behave like, the bone marrow V-able line to behave like a bone marrow pro-V cell. Can you restore contraction? And if so, does it make Vs accessible for recombination? And again, upstream of this locus, you've got CBEs everywhere. And I just have to mention, they would impede scanning no matter what, unless something somehow neutralizes them. So that was the experiment. If we neutralize CB impediments across the whole chromosomes, would we get rag scanning? And we did that by using a standard inducible MAI-made Degron approach in the Abelson lines, where you basically target the RAD21 component of COHESN for degradation. You basically can get rid of all of the COHESN. Really, I mean, all the COHESN in this experiment, I'm going to get into the CDCF. This was the control experiment. We did it. We got rid of COHESN. It basically blocked all recombination except for that D that I told you gets in by diffusion and sits in the recombination center. That recombined very well, and it was a control that getting rid of COHESN didn't stop VDJ recombination, but it stopped locus contraction completely and all VDJ recombination beyond that D in the recombination center. So that's in this paper. But then what Xiaoqing and Jinglan did was to deplete CTCF. That gave a very striking result. So here again is the parental lines, and so we got rid of the Ds right now, but they do little VDJ rearrangement and rearrange just a few of the proximal Vs. Once CDCF was completely degraded into the cells, their relative level of VDJ recombination goes up to that nearly like a bone marrow pro-B cell, and their pattern of rearrangements of Vs across the locus becomes highly significantly similar. It's not identical, and there's everyone to get into the reasons why that is. It's complicated, but it clearly it's very, very similar, and it clearly extends across the whole locus. And the level of VDJ recombination is bone marrow levels. So that finding shows that down modulation of CBE impediments activates locus contraction and promotes rag scanning to distal Vs. And it provides proof of principle, it's not proof of principle, that developmental modulation of CBE ability to impede scanning and or modulation of somehow ability of COHESN to get past scanning impediments, one or the other, could serve this function in a normal situation. So that's where we got to with the able line. So the next thing, which I didn't talk about last year in depth, I talked a little bit, we needed to address was whether rag scanning really is involved in primary bone marrow cells, and if so, what is the mechanism? Okay, so back to this slide I showed you before from Case Murray. It's the idea of the rosettes of locus contraction and the idea that all the V's are organized to join deletionally, so you can't learn much, or you can learn something, but not as much as you might hope by what I'm going to tell you. But all these cryptic RSSs are in both directions, so you can find out what happens to them, and that really gives you the clear answer in terms of whether something's going through the locus or something's diffusing unidirectionally. So this work done by Hai-Cheng Dai, Hongli Hu, and Jingman Liu, and it's testing in bone marrow cells. And what Hai-Cheng did, and was worked together with the other two, was in a sense a simple experiment. He basically in ES cells inverted the entire IGH locus, and yet Ralph, we did every control to make sure we didn't have any. We actually delete in some cases the other chromosome. This came from a thesis committee yesterday, and what he did was left one V intact, 81X. But everything else is completely turned in the other direction. And then he asked, what does that do to utilization? So that you look at utilization. Here's 81X, always used a lot. Here's everybody else. This is in the bone marrow. Inverting the locus basically killed the utilization of every V in the locus. So they're now pointed in the opposite direction, and that would be totally consistent with a scanning mechanism. You could think of other mechanisms, so we did more experiments. But here's another point, locus contraction. So to look at the influence of inverting this 2.3 megabase inversion, he looked at the interaction of the recombination center, which is for us the key thing, because that's the downstream block where reg scanning sets up and pulls the rest of the locus through it. He asked how that interacts with the rest of the locus. And here it is in wild type cells. And you can see there are a series of big interaction peaks, and these are correlate with transcription, with residual CBE activity, and other things. And they're hallmarks of locus contraction, the way people would describe it. When we invert the locus, it is amazing. It is a mirror image. Nothing goes away. All the same peaks stay there. Same peaks. They're just in the opposite orientation and direction. So we haven't affected locus contraction because everything is still interacting just as it did with the recombination center. Yeah, a little more or a little less, but it's all interacting. We look at also at CBE interactions, and interestingly, those are not nearly so much affected, even though their orientation also gets changed. It's in these papers. So that was a little surprise to people, but I think not to us, because we think the key player in this is not a CBE with respect to the recombination center. That's the RAG, JH, enhancer bound region, our interpretation. But here's the experiment, I think, that proves this. So now what we're not looking at is the recombination signal sequence, true ones. We throw those all out, and we look along the locus. You have CAC, 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 that's lined up pointing at the D, and you have GTGs, they're lined up. Go back. How that happened? This jumps on me. Come on. Went the wrong way. Yeah. So you got the GTGs, and they're in the opposite direction. So basically, these guys will rearrange by inversion. And if you bait from the recombination center, you get the D's area. Those are also cryptic sequences there, but you get across the entire VH locus a series of peaks. And that's all these CAC or various types of cryptic recombination sequences that are pointing convergently. So it's what you expect, almost nothing in the other orientation. Now, here's the remarkable thing. If you invert the locus, so now all these things that were CACs or become GDGs, and the GDGs become CACs, you see, and now it's just shown to say what's rearranging. It's not showing it really inversional deletional, but it's showing sequences that were in the inversional orientation before, now rearranged by deletion, and they rearranged quite robustly. So by inverting, you're finding all these cryptic RSSs that previously were in the wrong direction. But interestingly enough, when you get to the right direction, that's at the end of the IGH locus. Now, because you don't have the normal RSSs dominating, scanning keeps going, and it keeps going, except it goes back to the right orientation, and it goes all the way to the telomere. It never stops. It goes through about 5 convergent CBE domains. So there's something really, I think, was amazing to us about this is Upon inversion of the locus, reg gets the cryptic RSSs that are now in the other direction, but it also gets the cryptic RSSs that are upstream, which means it's continuing upstream. And that to us says that CBEs are deregulated universally because these have no relationship to these. So it's direct support that the mechanism may be a major down regulation of reg scanning. So we looked for a factor that could be involved, and the first thing we did was to look for COHESN complex factor that might be low in expression in bone marrow cells, pro-B cells versus ABLs, and one stood out, WAPL, I'll go to it. Its transcription is really low. Its expression is very low compared to what's in V-ABL cells. Details in the paper. Again, Cheng Li, I didn't mean to put Tom Look and hit it in here for any particular reason, except that's the only good one I had, Cheng. So he initiated this last experiment with Haicheng and Hong Li. And that is, okay, we now say this WAPL, based on our down expression in the bone marrow, could be a key factor. How can we prove it? And the easiest way for us to do it, and I say Manrad Busslinger just had a paper, a contemporaneous one, different angle, not always the same conclusions, but both conclusions that WAPL was about this role for WAPL. And the idea is WAPL is the release factor, wings like apart, and loads COHESN by transiently opening the ring. And that contributes to all, a lot of work on this protein, its role in regulating chromatin structure and segregation. It's been known that stable depletion of WAPL is cell lethal, but it's not lethal in G1 arrested ABL lines, which meant we could study it. And it's known by studies of Busslinger, not the Busslinger I'm talking about my contemporary, I'm talking about his son when he was working with Jan Michael Peters next door, showed the COHESN's position in genomes by transcription, CDCF, and WAPL. And if WAPL depletion while the cells are alive extends the size of loop domains. So it has the right kind of properties. So we degraded it in ABL lines. I won't go through all the details, but we degraded it to the point that it was undetectable. And what did it do for loop extrusion? That basically the treated V-ABL lines, the green dots versus the blue, look very much like bone marrow cells in terms of their peaks. It got much higher density of these high-level peaks. So it looked certainly like it activated loop extrusion. There's always a low level in these lines, but highly activated. And correlating with that, it activated VDJ recombination across the whole length of the locus, whereas there's nothing in the parental lines. So it clearly can activate loop extrusion and locus contraction, and all the correlates with the bone marrow say it's probably the key factor. We also did experiments where we've only inverted part of the locus, and of course that part doesn't rearrange, and then we inverted the whole locus over again, then that's the only part that does rearrange. So I think Haicheng's done every experiment you can imagine to really try to nail this. Last point here is that when you have when you have untreated parental cells and you use a recombination center as bait, there's not much interacting outside. When you use the recombination center as bait in WAPL depleted, lots of peaks. And then if you use the peaks as bait, they can see other things like this peak, can see other peaks out here. Some are V's, some are other sequences, but it doesn't see the recombination center. But when you deplete WAPL, its major interactor is the recombination center. Same for that peak. So I think that the point is that downregulation of WAPL allows these sequences and the distal part of the V locus to interact with the recombination center. And when they have recombination signal sequences, they can get rearranged. The model, good. So this is a new model, and we've had a lot of different kinds of models, and it takes into account a couple of other findings we've made along the way that we think are incredibly interesting potentially, and they're on our horizon as something that we really want to follow. So data that came out of Yu Zhang's paper, particularly when she did the dead Cas9 blocking of extrusion and scanning, implicated a recombination center that had bound RAG couldn't extrude DNA or chromatin passed it, as well as one that didn't. And then over the next couple of stories we've had here, more and more evidence that says that you can extrude further if you don't have that big, gigantic, bulky rag bound there. You can still extrude with it there, but not to the same degree. So the model that we're kind of thinking of, we're seriously thinking of, is that the one way to think about how this could occur is sort of a combination of loop extrusion and also loop extrusion mediated locus contraction and scanning, so that the two things that fit in a certain way that makes sense is that in progenitor B cells, everything we've done, where I show you interactions, it's done in RAG deficient cells because you don't want to have recombination occurring there. And then you get some discords, like I've mentioned. But we think that RAG, if you had a set of progenitor B cells at different points in their history and their cycle and everything else, that they could have a lot of cells that are scanned to different positions along the locus in the absence of RAG, which will allow scanning, based on what we've seen, to go a lot further. But it would be all the way around, depending on what the timing was of initiation. If at some point RAG gets turned on, what it means is RAG, yes, can bind to the recombination center at that point, but it could also bind to the recombination center at that point, or that point, or that point, which means that extrusion would have compacted the whole locus and done all these things, but the scanning part would be still RAG binding to the recombination center and being able to pick up the nearby sequences mainly, I say mainly, by extrusion because of the linearity of the whole process. Now, can diffusion happen? They're oriented in the right direction. If you got close enough, we have it in our papers. If you're within a linear, say 20 kb, then you can actually diffuse. And if you're in the right orientation, you'll get rearranged. So that could play a role, but it has to be a more minor role since we're seeing extrusion going through both sides, seeing sequences on both sides of the whole locus. So that's our thinking of the model. And the last thing is it's not over by any means. I mean, it just gets more complicated. So remember I said these same able lines that have not contracted their heavy chain locus, don't rearrange distal V's, have moved on to the kappa locus. And here is what their rearrangement patterns look like. So kappa is in a locus that it has a J, has some enhancers, theoretically a recombination center. It's got some elements that look just like, in a certain sense, like IGCR1, two convergent sets of CBs, separating the J's from the V's, but the V's are really different. They're kind of mixed up in this part of the locus, the proximal part of the locus. Some are in convergent orientation, some are in divergent orientation, or in the same orientation. So oriented to only be rearranged by inversion. Big cluster of ones that are rearranged, oriented for inversion, RSS going the wrong way, and then some at the other end oriented for deletion. So kappa, but here, baiting from the J kappa region, what you can see, deletion is above the line, blue, inversion is below the line, red, and they're all getting rearranged to some degree or another. So it's figuring out how to do it, and it's not linear exactly, that's for sure. And it's going across the entire locus of a cell. It can't, its WAPL levels won't permit it for the heavy chain locus. So it means, it could be something else involved that's totally different, or just a different version of the same story, but maybe some minor changes in WAPL levels. This locus has fewer CBEs. I mean, you can think of a lot of interesting things, but they're all testable. The only one thing we know is if we completely get rid of WAPL, we get a pattern where now you get more rearrangements of the distal V's, and and less of the proximal V's, which fits with this idea of the loops getting bigger. So it's responding to WAPL, but just not in a dramatic way. The other thing we know from Jingmon Liu's recent work, do the same story with the kappa locus. Just cut it and invert it, the whole locus, intact, and say, does it have any impact on any rearrangement pattern? The bottom line, which, maybe these aren't plotted exactly the same way, but it's basically showing that when you invert it, what was distal is now proximal. They get arranged now. They still get rearranged. In this case, by deletion instead. Inversion in the middle. Nothing. It's now deletional joining, whereas before it was inversional joining. But the frequency isn't changing very much. So it's saying the kappa locus is working under very different principles. We do know that we can make proximal V genes rearrange by deleting the IGC or the sort of cis element. We've used those Silu more recently in some of our HIV and antibody models. They're very convenient. So it can scan, but the question is, how does it do all this by scanning? There are scanning models one could imagine that would explain it, or maybe it's not using that exact same mechanism. We will see. A lot of things are going on. So, but we're looking at all the why are these so different, and then it's the combination centers are different. We know in some ways, chromatin structural elements could be expressed differently. Maybe it's something different than WAPL for modifications of COHESN complex acid, but clearly the chromosomal organization is going to be changed here. And I think that how that works to bring everything together will be equally interesting. Okay, thanks a lot. I think I mentioned almost everybody. Eddie Dring, Nia Christus, and Jeffrey Zarita, the computational people. Eddie worked closely in the lab as a tech with Chao-Ching. They've all now moved on to new positions. And we've had some collaboration with Rafael Casellas and his lab, and we had a collaboration early on with Erez Lieberman Aiden, always a very fun person to talk to. Yeah, I think that's it, Ralph. I'll let you a minute maybe.
Fantastic. Thanks so much, Fred. What a wonderful presentation. We've got a few questions, and I think we have time for some of them. So our first question is from Diogo Maia Silva. Can you comment on the time scale of these recombination events? Do cells ever divide while RAG scanning is happening? And if so, does RAG restart where it left off?
I'm sorry, what, did you say that again?
The question is, can you comment on the time scale of these recombination events?
Yeah, I don't think I can comment. Other people have commented on them in the bone marrow situation a long time ago, and I think they could probably have it down to in a period of days at a time. I think in our Abelson cell lines, events start happening as soon as we put them, as soon as we induce them. So in some cases, we're just adding RAG to them. So we're finding these things within a day, but I think in a much shorter period. So there'll be some accumulation over that time. It's an exceedingly interesting question to us at the moment, simply because of what I was saying, this model about the timing of RAG versus the timing of extrusion. It's something we want to figure out. I should mention I've got a graduate student also, Yuan Zhang, who's doing single cell work with Xiaowei at Harvard. And maybe we'll get a lot of more time scale types of issues covered with a lot of these events in the future.
Great. A question from Benjamin Gewurtz. He's wondering if there might be also a role for DNA methylation in regulation of CTCF in VDJ recombination.
Of regulating
CTF sites, that is, perhaps.
Yeah, I've heard that mentioned before, and it was actually mentioned at a conference I wasn't at, and the answer to that was given is we're working on it from the postdoc who is involved. It's clearly something that would be of interest in looking at. Right now with the bone marrow, we're more, the WAPL thing seems to be a more likely culprit based on everything I've shown you. And also Meinrad Buschlinger's lab works next to Jan Michael Peters' lab, and between the two of them, they've done a lot of studies. Well, Meinrad in particular, PAX5. So he thinks his argument that he published recently is that PAX5 actually downregulates WAPL, and that is how. And then we've known for a long time that PAX5 somehow contributes to locus contraction. So he's proposed that that's the way it goes. So it looks, if you connect all those dots, it looked like a pretty strong case. But for the kappa locus, for other factors, and everything's not perfect. I mean, it's very close, but not quite. So I think methylation of CDCF sites is still something that we need to look at along with other things, or somebody's going to look at them.
Right. Another question. Does the V lambda locus rearrange in the same or by a different mechanism from V kappa?
Yeah, that's another good question. We were working on the lambda locus pretty hard until about 2015 when we discovered this whole scanning phenomenon, put more work on the IGH locus. Up until that time, Xiaoqingba, if I can remember, until he jump ship from lambda locus. He has a very interesting story on the lambda locus to finish, but the lambda locus in mouse is whoever asked the question probably knows is very simple. It's only got like a couple of J's and a couple of V's. They are themselves in separate loop domains and potentially separately regulated, as best I can remember. In humans, it's a totally different story because you've got many V lambdas and C lambdas and J lambdas. I think they still, and their organization, I just don't think the lambda locus, to my knowledge, in humans has been studied carefully enough. It's a little harder because you don't have Abel lines or things like that. You can't, it's just harder, but it won't be the same as kappa. It doesn't rearrange at the same developmental time. And whether there's communication between the two is a really interesting story that goes back about 30 years. I don't think we know the answer to that yet either. So I think all these loci, T cell receptor loci, lambda loci, they're going to incorporate these general mechanisms to one degree or another, maybe like IGH and lambda and kappa, maybe not exactly the same way, but variations on a theme. That's what I would guess.
The question about the stalling of RAGs at CBEs and how that tends to promote selection of a particular segment for a combination. Is the stalling, is it at all clear whether the stalling itself biochemically activates RAG, or is it thought that stalling is, it's really just enhancing the probability of recombination occurring between the segments?
Right. So I would say the story is too young to give you a definitive answer to that question. I'd say the bias, at least my bias, is that it is more of having two sequences in close proximity for a longer period of time. So we're talking about diffusion or getting closer. I mean, you're sitting on top of each other. So I think if there's some fine tuning to do there to get embedded into a site, it's given a much more chance for that to happen. Could there be some chemical activity going on there that would change RAG activity or something? I mean, I don't think we could rule that out. I think a lot more work would need to be done.
Right. And a similar question actually about WOPL, that factor is clearly influencing the efficiency of traversing of CBE sites like COHESN. So are there other COHESN regulators that you think might be involved in addition to WOPL? And might there be signaling events that activate or inhibit them at specific CBEs?
It's all possible. I mean, so we started with two obvious, get rid of CDCF and get rid of the CBEs, or get rid of WOPL and allow, what sort of the two ends of the spectrum of how, if there was a mechanism, those would be one of those two mechanisms would seem likely. Now, could it be some of the other factors? Absolutely, because they're involved in allowing WOPL and CDCF to do what they do. So I think there's still others. There are potentially other players. It's just, I guess the WOPL role for the IGH focus seems so strong. It diminished the. I had a number of people working on the other factors, take them one at a time and enable some lines, right? And some of them I have no doubt will give very similar phenotypes. Then it's a question of how do you prove then that they're doing that same role. So I think the WAP will diminish that activity a lot. I think it'll come back because I think we haven't, it's not all solved. And so I think, paper reviewers always say, hey, why don't you look at this factor, or my favorite factor, or whatever. I agree. It's clearly there's other players there, a lot, and there could be other points of regulation.
Yeah,
and it may not be the same, and it's very clear that it's probably not the same in every locus. Some of the TCR loci don't even contract the same way, and some T cells that are undergoing contraction and rearrangement that express high levels of WAPL. So it could be done differently in different cell types.
Yeah.
And I suspect there'll be a few people looking in their own favorite cell types. For us, I think the heavy chain locus, the kappa locus, and if we can get to the lambda locus, that'd be more than enough to keep us busy for a little while, because there's still a lot to do.
Yeah, I think you're going to stay busy, Fred.
I
think you're going to stay busy.
Yeah, try to.
Okay. So, Fred, thanks so much for a fantastic seminar. Very, very lucid and thought-provoking. So thanks again, and we will see you all for the next DFHCC Science Symposium.
Thanks, Ralph.